pleasure to introduce Dr. Shahar Keinan. Dr. Shahar Keinan is the co-founder and CEO of Polaris Quantum Biotech. She has over 20 years of extensive experience in the field of computational and theoretical chemistry and has published over 50 peer-reviewed manuscripts in the fields of in silico drug design and discovery, as well as molecular materials design and computational methods development. Shahar has received a PhD in theoretical chemistry from the Hebrew University of Jerusalem, after which she moved to Northwestern University and Duke University for postdoc positions. Previously, she was CSO of Cloud Pharmaceuticals, and she has been instrumental in the development of the quantum molecular design process since its inception at Duke University. And she has pioneered the use of computational chemistry algorithms to optimize electro-optical materials and small molecule drugs. We now welcome Dr. Keinan to present her talk entitled A Quantum Revolution in Drug Discovery.
Thank you, Dr. Andrea, and the rest of the Precision team. I'm very happy to present here, and I'm looking forward to this to the talk and for questions from the audience. So, as Andrea said, I'm a CEO and co-founder of Polaris Quantum Biotech. And what we're doing is we are using quantum computers to usher in a quantum revolution in drug discovery. Okay. What we do is a rapid drug discovery in a vast chemical space powered by quantum computing. We have the first working drug discovery platform that is built and running currently on a quantum computer. And we use that technology to build a very large virtual chemical space, over 30 billion molecules, and find the right molecule in that virtual chemical space. Today, I will talk with you about how we do this, and I will give you an example from a white paper we published. Okay, before I do that, let me remind you that there are different kinds of quantum computers. You've all probably have heard about gate quantum computers. These are the ones that IBM and Google and Rigetti are developing. These are quantum computers that can solve all of quantum computing problem types. Things like encryption, quantum location, all of these kind of problems they can solve. Currently, there's only very small system, up to 32 qubits, although IBM says that they will get to much larger system in the next 2 years. They're talking about getting to 1,000 qubits in the next 2 years. However, there's another family of quantum computers, and these are quantum annealers. So quantum annealers solve only a certain problem of questions, and that's optimization problems. However, we can build and have been building very large system for that. So 4 to 10,000 qubits are available. There are several of these kind of quantum system available. There's one with Fujitsu, which is a digital annealer. It's not similar to quantum annealer, but it can solve the same problems with the same speed. D-Wave has a quantum annealer and a very friendly set of examples that people are really encouraged to go and try solving some of their optimization problems there. These are quantum annealers, and this is the system that we are using and solving chemistry problems on that. Mm-hmm. So what's the problem we're trying to solve? Currently, drug discovery processes are lengthy and expensive. Everybody in this conference knows that. But we also know that we know, or the industry knows, how to design molecules on the computer. We know how to look at small changes in scaffold, changing a hydrogen to a CH3 group, and seeing how those reflect in activity, in binding affinity. But we know the industry knows how to do that only on small number of molecules, 500 to 1,000. So what the process in the industry right now is you design some molecules on the computer using small changes to your scaffold or to molecules you got from high throughput screening. You do the synthesis, you then do the measurements. You learn something, you definitely learn something. And then you go back to the drawing board. So you do these 3, 4, maybe 5 times before you have a set of molecules that are drug leads, that are good enough to go to more testing and maybe move all the way to testing to toxicity and testing in human. This can take up to 3 years, upwards of $4 million. And that's because we do these multiple cycles and we do many more synthesis and measurements than what we should do. What does Polaris do? Instead of doing these kind of cycles, we do this only once. But how can we do that? We do that by looking at very large chemical space. So instead of every time looking at a couple of hundreds or a couple of thousands molecules, we design a virtual chemical space that is relevant for the specific protein you're interested in and the specific disease that you're interested in. And we are using quantum computers to find the best molecules in that very large chemical space. Instead of doing multiple cycles, we do only a single cycle. And instead of doing this in 3 years, we do this only in 4 months. Because of that, we find the same molecules. We find them faster and find them very much cheaper than what you will get today. And because we are looking at much larger solution space, much larger chemical space, we can look at new chemistry, new scaffolds, new things that you maybe haven't checked before or haven't looked at before. So we published a proof of concept. This proof of concept was done with the Fujitsu Digital Annealer and the Fujitsu Digital Annealer team located in London. And here you have the address for this white paper. And what we've done there is we've chosen a target that the industry have worked on before, dengue, the dengue virus, RNA-dependent RNA polymerase, RDRP. And we've done exactly what I've said we will do. We designed a large billions of molecules, and this is fragment-based chemical library. We used the Fujitsu Digital Annealer to distill the library to about 3,000 molecules. And that kind of calculation takes under 5 minutes to complete. You have a library, you build the library, you build your set of constraints, what are kind of molecules you're interested in, and it takes a quantum computer about 3 minutes to find the best molecules in that library. We targeted the dengue fever. It's a mosquito-borne tropical disease. It is caused by the dengue virus. And although in the last year and a half almost, we are only talking about one kind of viruses, dengue is still a problem. In many places of the world, it is a tropical disease. And the problem with it is that most people, when they get it the first time, it's a painful disease. It's high fever, muscle, joint pains, sometimes rashes, but most people recover from it. But in some cases, this disease can become a hemorrhagic fever. So it is lethal. And there are 4 kinds of dengue fevers. And if you got sick with one kind of dengue fever and then you get infected with a second kind of dengue fever, the proportion of people that do not survive the second time is much, much higher. There is no drug or medicine for the disease. There is vaccine, but it is recommended for very small number of patients, and it has it been taken out of the market in several of the large markets of dengue. So we thought this is, when we started working on this, we identified dengue as our target. And one of the reasons that we were interested in that, I'll go back to those slides in a minute, is because we identified a specific protein in dengue, which is the RNA-dependent RNA polymerase. And this is part of the dengue virus non-structural protein, 5 or NS5. And this protein was studied extensively by Novartis, and especially their team, their tropical disease team that was located in Singapore. And they discovered a conserved pocket in all of dengues, and they used a fragment campaign. So this was something that we were interested in, and it was a good match for our technology as well. They published 4 manuscripts, each one, every year they published 1 manuscript, and they started again by discovering the pocket, looking at fragment campaign, but a year later they found that they can extend the length of their molecules and remove one of the water molecules that were occupying that pocket. And the ligand activity increased by 1 to 2 orders of magnitude. And we were interested in that because this is a different geometry. However, it's the same binding pocket. So can our method, can our library find those kinds of molecules? So before I do that, let's go back to the process that we're using. Our platform is called Tachyon. And what we do is we start from a biological target. In this case, it's the dengue RDRP protein. We start from a biological target. We build a very large solution space of billions of molecules. That's the molecular library. We search that molecular library at 2, sometimes 3 different levels of calculations, of accuracy, and the number of molecules that we can deal with. And I'll talk about that to find the right molecules. And then we synthesize and measure the activities. This is an automated, it enables upscaling of the process and, of course, parallel project execution. So we can do multiple projects at the same time. So let's start from a biological target. I talked a bit about the dengue RNA-dependent RNA polymerase. And here, by the way, is the Novartis manuscripts. So after we chose the target, we started to build the molecular library. I'm not going to give you here the molecular library because we're still working on this project, and we are these molecules are being synthesized now. But when you can build that virtual library in 2 different ways. The first one is you start from a library of commercially available compounds. Enamine has that kind of library. Wuxi has that kind of library. There are others. They are 1 to 1.5 billion molecules. Wuxi, I think, is a bit bigger. And then you take those and you reformat those to make them optimized for search on a quantum computer or quantum annealing platform. That's one way of building your library. The other way of building your library is by designing a specific, targeted, diverse chemical space library. And for example, you can look at the example here. This may be something that binds to a kinase or a group like that. And you can replace each of these Rs here with a different fragment and build a very large library. This is for cases where the commercially available library is not going to be good enough because you already know that you don't have good molecules in it, for example. Or if you're looking for molecules that are either going to be charged or non-charged or have a very specific protein environment requirements. We include, of course, synthetic accessibility, so we make sure the molecules that we design are, of course, synthesizable. And after you build that library, you need to translate the library and the chemistry to the language of quantum computing. Now, on an optimization problem, when we solve on a quantum computer, we write this as something called QUBO, quadratic unconstrained binary optimization. This is the kind of problems that a quantum annealer can solve very easy and very fast. There's, if you know, icing kind of problems or traveling salesman kind of problems are also easily translated to QUBOs. What we need to translate here is both how do you translate the chemistry itself, so which group is, how do you represent it inside the optimization, but also what are the constraints that you're looking for, and what are the penalties, and what are the incentives. So what kind of groups you want to have at a certain position, what you don't want, and what are your constraints. Are you looking for molecules that have a certain molecular weight, a certain ADME properties, these kind of things. So we divide this into two, physicochemical constraints and structural constraints. And both of these are part of what build the QUBO. We evaluate each molecule. Again, this is a quantum computer, so we can evaluate each molecule. We don't need to use some kind of a sampling technique here. And the optimization goal here is exactly to find all the molecules in the chemical library that match the criteria. So if we are interested, for example, here, we're interested in certain distances between the fragments of the molecule. This is something that we will add as a constraint to the QUBO. And then we will search for those constraints. And only molecules that have this kind of distance, this kind of distance, this kind of distance, and maybe this kind of angle will be chosen. And of course, that also means we have the physical chemical constraints. So a certain molecular weight, a certain SASA, and all the rest. Mm-hmm. So after we built that molecular library, we're then going to search that molecular library. We're using both quantum computing and QMMM. We will start with the quantum computing, and this example was done on the Fujitsu digital annealer that we've used. They have, when we ran it there, they had about 4,000 qubits. Right now, if you work on that machine, you have access to 10,000 qubits. It's the largest in the world right now. And here are some of the results that we find. We are comparing here random molecules that you can choose from the library to molecules that the annealer found. You can see that we find molecules that are 2 orders of magnitude better than looking at random, and we've looked at other ways of finding molecules, and we are always getting better molecules. This is a log scale. Okay. And what is written here as energy is basically a multi-object optimization function that include both the physicochemical constraints and the structural constraints. So this is not real energy. Some properties that we calculated during the run, and we want to show that the molecules that we find there are good drug-like properties. Here is the distribution of molecular weight. We had a cutoff of 500. This is synthetic accessibility score. Again, we wanted to make sure these molecules are accessible, and log P of the molecules that we designed here. And this is about 3,000, the top 3,000 molecules that we've looked at here. We then take these molecules, and as you can see, probably the quantum computing can look at very large chemical spaces, but maybe the level of accuracy is not as high as we would like it. And then we go through QMMM calculations, which are more accurate. And we are using the QMMM with the linear interaction energy formalism. This is an LIE formalism. QMMM is a multi-scale, multi-resolution calculations. It includes fully atomistic, so all the water molecules, all the correct protonation states. We are sampling both the protein conformation, the ligand conformation in the pocket and in water, and of course all the cofactors. This is also, and if anybody is interested in that, we published, and this is from the previous company, we published some work on that. It is also embarrassingly parallel, so if you want to run it on multiple cloud environments, you can do that. Here is some of the results. This is the paper that I said, comparing measured experimental IC50 to calculated delta G for 4 different proteins, 2 sets of ligand per each one of them, and you will see here that you can compare different proteins if you're interested in selectivity calculations, for example, and also you can compare different ligand structures. So if you're interested in getting out of your local scaffolds and trying completely novel scaffolds. The LIE QMMM calculations and methods have been used before to design molecular drugs. And if anybody, this is a published work, so everybody's interested in that can find it out. So after we've done the quantum computing and QMMM, we choose the molecules for synthesis and measurements. This is where we are right now with the dengue published proof of concept. These molecules are being synthesized. The first thing, of course, when we got all of the results out, the first thing we did is to see whether or not the molecules that the quantum computer chose has the right geometry, whether or not they try to remove that water molecule extending the original scaffold. And what we found is about half of the molecules that we looked at, including that extension, that R group that was much longer than the rest of them. So we can see how this kind of methodology can find novel scaffolds and novel positions inside the pocket. And we've designed about about 20 other molecules that we chose for synthesis, and some of them will start synthesis right now. I want to leave some time for questions. So let me just finish here by talking about the team that did a lot of this work. Myself, you've met me already. Bill Shipman is Polaris co-founder and CTO. Worked a lot before in drug discovery, drug design, and in genomics. The senior computational chemist is Ken Byler, and we have 2 industry experts. Both of them are called Ian, that supports us, both in Ian Reynolds in pharmacology and CNS, and Ian Cloudsdale, who is the medicinal chemistry expert on our team. The process itself starts from a target, a biological target, a protein, and the definition of what you're interested in. What's the molecular weight? What's the toxicity profile? And what's the ADME profile of the molecules? We build the library, we search the library, and in 2 to 3 months, a set of 10 to 20 candidates. We found a set of 10 to 20 candidates. I am going to stop here and answer questions if there's any.
If there's any questions, I think Mr. Guru Singh, I think you had a question for her. If you want to type it in the QA or discuss box, feel free. Just take a moment and have the audience type it in.
Sure. And let me just add here that I'm on LinkedIn. You have my emails. I'll be happy to connect with people and answer questions as well when this seminar is finished. And we are looking for collaboration and interesting projects, and as well as service projects. Both of these are something that we're interested in pursuing at the end of this conference. Mm-hmm.
We'll just give another minute or so. Yeah.
I just had a quick question. So obviously the world is thinking about other viruses like coronaviruses. Has this computing been used to potentially look for new therapeutics for COVID type infections?
Yes, so we are collaborating with the Fujitsu team on COVID as well. This is we have preliminary results there, but there's quite a lot of published work, computational published work on COVID that is not supported by experimental results, and we decided not to muddy the water some more and only wait for when we have experimental data to look at that.
Oh, very
to publish
cool.
that. Okay, but it is of course a target that we've been looking at. It's of course.
And there is a question.
I'm glad.
Yeah.
And there is a question that just came up from Dr. Frederick from the University of Toronto. He's saying, "Hi, Dr. Shahar. How do you handle selectivity, i.e., kinase inhibitor selectivity?"
Hey, sure. So we do this in two different levels. In the first level is when we design the library, we're saying, okay, let's compare the binding pockets of the two kinases. And we're not just looking at the small area around where the 3 amino acids are that you bind for, but let's look at a bit further away and design the library in a way that will have a preference for one kinase versus the other. Okay, so if we know that, I'm comparing here two pockets. One is doesn't have that location. The other has a location that you can bind for. So then we will make sure that all of our ligands will have the possibility to bind just for one kinase and not to the other. That's the first thing. The second part is that when we finish or when we do the QMMM calculations, we are calculating the ligands versus both kinases or versus several kinases. And that's the QMMM calculations are accurate enough for that, to be able to predict selectivity. And some of the work that was done with QMMM with the LIE was to design selective JAK2 inhibitors, for example, that don't bind to JAK1, JAK3, or TYK2, but do only bind to JAK2. But also there was a work about designing inhibitors that are pan-JAK, so bind to all four members of the JAK family. So you do it in two different levels, a very broad level and a much more accurate, high cost, but also high accuracy calculation at the QMMM level.
Cool. Yep. Thank you so much again, Dr. Shahar, for your amazing presentation. And as she mentioned, you can connect with her on LinkedIn via email as well. And
Thank you very much. And I'm looking forward to the rest of the conference.
Absolutely. And with that said, we'll head over to our next session, which is a tag team presentation from Rapid Novar. So let's all head over.